to the Temporal Genomics seminar, and happy Earth Day, everyone. Thanks everyone for coming. My name is Brendan Reed, and I'm a postdoc in Malin Pinsky's lab at Rutgers University in New Jersey. I'm speaking to you from the traditional territory of the Lenni Lenape people. This series is organized by the Research Coordination Network for Evolution in Changing Seas Temporal Genomics Working Group. If you want to learn a little bit more about our working group, we have a lot of information on our website. I will link to that in the chat once I finish my little intro here. But this is a bi-weekly seminar that's aimed at showcasing research into temporal genomics. And by this we mean studies that use genetic samples collected over time, often historic and contemporary genetic samples, to examine questions relevant to the ecology and evolution of species over the past several hundred years, and especially their responses to human-induced changes in their environment. We're also really interested in highlighting best practices and new methods for those of us who are interested in doing temporal genomics work themselves. We have a diverse range of topics and speakers planned for the seminar series, and we hope that you'll stay with us over the course of the next few months. You can see the schedule on our website too. Yeah, for our next seminar on May 6th, we will actually have the first of 3 roundtable sessions where we have 4 early career researchers giving shorter presentations on their work. So that one should be really neat. Be sure to check that one out. Also know that we don't have enough time in the seminar series to highlight all of the amazing work that people are doing in temporal genomics. So we are planning a Twitter conference where even more people will be able to showcase their ideas. Details for that are forthcoming. We're working those up now, but if you'd like to participate, stay tuned and we should have more concrete details pretty soon. But yeah, just a few ground rules. While our speaker is presenting, we ask you to please be respectful, keep yourself muted, hold all questions until after the presentation is over. Once the speaker is done, we ask you to either use the raise hand button or indicate in the chat that you would like to ask a question. Katrina Catalano is helping me moderate. Either she or I will call on you in the chat, then you can ask your question. Also note that we do have a lot of registrants. If something unforeseen happens, if Zoom crashes or something, or if there's something else, we do have an alternate webinar link that's available. We'll do our best to email that link out to all of you quickly, and then we can resume in the webinar if we have to, but hopefully nothing will happen. Okay, so now that all of that is the way, I have the great pleasure of introducing this week's speaker, Dr. Evelyn Jensen. So Evelyn earned her bachelor's in science and evolutionary biology at the University of Alberta. She worked there with Sally Lays and David Coltman on an honors thesis on the genetics of sponges. She did her master's in science and her PhD in biology at the University of British Columbia, Okanagan, where she worked with Michael Rossello on the genetics of Western painted turtles and Galapagos tortoises. And after UBC, she was an NSERC and Genome Canada postdoctoral fellow working with Stephen Lougheed at Queen's University, Ontario. And then she followed this with a fellowship with another postdoctoral position at Yale with Gisela Caccone. And just recently, she's accepted a lecture position, which is basically a tenure track assistant professorship at Newcastle University in the UK. So congratulations on that. And yeah, the floor is yours, Evie. Take it away.
All right, well, thank you for that. I'm very excited to have the opportunity to talk today about some of our work with Galapagos tortoises. I could talk about tortoises all day, every day. So today is a special treat as far as I'm concerned. So please, if you have any questions or anything, I'm happy to chat at the end. Just trying to get you guys where I can see you without you being in the slide. Not going to happen. Oh, well. So I've titled this talk Temporal Tortoises, What We're Learning from Historical Specimens of Galapagos Giant Tortoise. So I'm going to tell you 3 different stories about different species of Galapagos tortoise, and we've learned different things using museum samples from all of them. So the very first thing I'm going to do is just acknowledge all of the wonderful people I get to work with on tortoises. So the PI at Yale, Gisela Caccone, research in her lab over the last 20 years has really given us everything we know about the evolutionary history and conservation genetics of Galapagos tortoises. And my PhD advisor, Mike Rusello, was once her postdoc. And so that's how, with him as my PhD advisor, I got to do a PhD on tortoises. And then I got to go do a postdoc with Gisela myself. There's also a long list of other collaborators there that have taken leads on different parts of the Galapagos tortoise sort of research story. And I'm very lucky to have such a wonderful group of people to work with on this research. So let's take a trip to the Galapagos. The Galapagos is a volcanic archipelago located exactly at the equator, about 900 kilometers off the coast of South America. It's formed over the last 5 million years or so as the Nazca plate is sliding towards South America. And this means that the western parts of the archipelago are younger than the eastern parts. The tortoises arrived in the Galapagos in a single colonization event, and from there they've radiated across most of the major islands, evolving into distinct species on each of them. So the islands to the east there, San Cristobal and Hispaniola, were the ones first colonized, and they had tortoises there before the westernmost islands like Isabella even formed. And so this is sort of a true radiation that follows the island progression rule, where younger islands have younger lineages. Typically, there is one species of tortoise on each island, with 2 exceptions. The first one is Isabella Island, which is the large L-shaped island here. And it is made up of 5 volcanoes that in the present day, with the present height of the volcanoes and present sea levels, they are connected into one island, but it does have these very separate areas of tortoise habitat. The other exception is Santa Cruz Island in the center here, which has 2 lineages of tortoise that arose through 2 separate colonization events. The Galapagos tortoises are famous for having 2 morphological types. There's your typical dome-shaped tortoise, as well as a saddleback form that has this large arch over the neck of the tortoise there. And this gives the tortoises a much larger range of motion. And it's thought that this is important for foraging as well as exhibiting aggressive behaviors. The locations where each morphotype are found corresponds to the environment there. So the dome type is found in lush sort of highland environments where there's a lot more vegetation. And the saddleback form is found in low elevation, arid places where it's thought this increased range of motion assists in foraging, as well as them being more territorial and sort of allow more aggressive interactions between tortoises. Unfortunately, through exploitation, the population of tortoises in the Galapagos has been reduced to an estimated 10% of historical levels. And although they're now protected and safe from most direct exploitation, 4 species have gone extinct, and all the rest of them are on the IUCN Red List. So some are vulnerable, some are endangered, and some are critically endangered. The good news is that since about the 1960s, the Galapagos National Park has been engaging in fairly aggressive conservation actions, from captive breeding to really extreme programs to eliminate invasive species. So these actions are helping to restore both the environment as well as the tortoise populations. I'm showing here a phylogeny of the radiation of tortoises involving all of the living and some of the extinct lineages. So this is based off of 3 mitochondrial genes. And the important thing to take away here is that the species are reciprocally monophyletic. So these are highly distinct, except on the island of Isabella, where in purple here in the purple clade, these lineages are all very recently diverged. And so they do not show reciprocal monophyly, as well as northern Isabella. Up here, this is called Volcano wolf. We'll talk more about that later. And Santiago, the northern part of Isabella was colonized by 2 different radiations from Santiago, and one of them was quite recent. So that's why they are not as distinct. But when we look at each of these populations using thousands of SNPs, so here DD-RAD of 10 individuals of each of the living lineages, we find that they are distinct in clustering analyses. I'm going to talk a little bit about the human history of the Galapagos, which helps build the story that we're going to talk about for the conservation and where the temporal samples come in. So the Galapagos do not have an indigenous population of humans. They were first discovered in 1535 when the Bishop of Panama was sailing up the coast of South America and his ship got blown off course and they landed in the Galapagos. Which nobody thought was a very nice place. And so they got out of there pretty quick. And it didn't really become a hotspot of activity until the 17 and 1800s, when the Galapagos became a very strategic hiding place for pirates between raids on the Spanish colonies in South America. Later on, it also became an important stopping place for whalers, taking advantage of the rich sort of seas for catching whales to the northwest of the Galapagos archipelago. And so at the Galapagos, these mariners found the tortoises to be an extremely valuable resource. And so there was massive harvesting of tortoises for their meat, basically. Tortoises being very hardy creatures, they can survive for months at a time without any food or water. And so they would fill the hulls of the ships with tortoises just stacked on top of each other. The tortoises would live like that. And then months into the voyage, they could slaughter a tortoise and have some fresh meat. So you can imagine what a valuable resource that would be. Unfortunately, the humans brought with them a lot of invasive species, everything from goats and pigs and cats to, importantly, rats. And rats, we know, are a problem on island systems, particularly all over the world. And the Galapagos is no different. In 1835, the Galapagos had its most famous visitor, Charles Darwin, sailing aboard the Beagle as they went around the globe. And it's his observations in the Galapagos that helped to formulate the theory of evolution through natural selection. And so I like to think of the Galapagos as sort of the conceptual home to evolutionary biology. Moving to the 1880s to the 1830s, the Galapagos really became of interest to natural historians and museums. And so numerous scientific expeditions were sent there to collect specimens to bring either live or preserved specimens back to Europe. And we know of at least 660 tortoise specimens that were collected during this time period. One of the most important expeditions was by the California Academy of Sciences between 1905 and 1906. They spent more than a year there and in the end collected over 70,000 specimens of animal to take back to their museum, including many specimens of tortoise that I will be talking about later. From 1959, the Galapagos became a national park, and surveys began to determine the status of the tortoise population. And unfortunately, by this time, they were quite decimated, as well as many other species. But this is where the conservation programs begin. So I'm going to focus for this first part specifically on Pinzon Island. Pinzon is in the center of the Galapagos archipelago. It's a very small island. It's only 18 square kilometers. And the species there is Chelinoidus duncanensis, formerly also called Ephipium. So some of the figures will see the word Ephipium. It is the smallest of the giant Galapagos tortoises. So they reach a maximum size of about 1 meter in length compared to the other species, particularly domed ones, which can get much larger. And because of their small size, they were relatively heavily impacted by harvesting because it only took one swarthy sailor to pick up a Pinzon tortoise and carry it off the island and onto the boat. Whereas these other ones, they were so big, they would require multiple people. And so the population was very heavily impacted by harvesting. In the 1970s, when they did surveys there, they found a small number of tortoises on Pinzon Island. So this is the top plot here. And this is just a histogram of the sizes of tortoises that they located. And the lower plot is another species on Isabella Island. And what you can notice is that the Pinzon tortoises, they found fewer of them. They're smaller. They don't get up to this, 1.5 meters in length. But importantly, there are no small tortoises at all. So the smallest tortoise they found had a carapace length of 50 centimeters. So very much a fully mature adult. And no juveniles or hatchlings were found at all. So although it was estimated that there were 150 tortoises on the island, there had apparently been no recruitment since about the 1890s due to the introduction of black rats that were eating all the hatchlings. So Pinzon was completely overrun with rats that would eat the hatchlings, eat the eggs, destroy the nests, all those sorts of things. So for most species, the story would end there because there has been no successful recruitment at this point for 80 years or so. And it's only because these are tortoises and they are so remarkably long-lived that there could just be this pause in recruitment, and then humans can get their act together and start doing something about it. And that's exactly what they did. So they started a Head Start program where rangers would go each year and collect eggs or juveniles before they've emerged from the nests, bring them to Santa Cruz Island to the Charles Darwin Research Station, and rear them in captivity until they're too big to be eaten by rats. So about 4 or 5 years old, at which point they would repatriate them back to Pinzon Island. And this program, at the time that I started my PhD, had been going on for more than 50 years, which really shows the incredible dedication to preserving these species and all the hard work that has gone into it. But there are some issues with this. And the key is that it's really not addressing the cause of the problem. So there were still rats on the island, and there was no recruitment happening outside of the Head Start program. And so if something happened and the humans stopped Head Starting them, the species would be in the same position as before, except, with a somewhat larger population size. There was also concerns mounting that because of all of this interference, there might be a skew in the population if they're sampling the same known nesting locations each year and perhaps are not getting a representative sample of eggs from all of the potential parents. So these are some genetic questions that we can address fairly readily. So we looked at addressing, do the Head Start generations have reduced genetic diversity? And has the genetic structure of the population been skewed over time? And to address this, we had temporal samples. So samples from the contemporary population, as well as samples that were collected by the California Academy of Science expedition back in 1906. So in 2014, we went and we collected as representative a sample that we could of all of the tortoises living on Pinzon. So you can see there the distribution of the tortoises we sampled, and this represents the full area of occupancy of the tortoises. The other parts of the island are not suitable tortoise habitat. So we used rad sequencing to discover and genotype SNPs for these 150 individuals. And then we took the sequences of those rad tags and developed those into capture probes with the help of the folks at MyBates. And so with our historical samples, which we had 78 from this 1906 collection, so it tells you just how successful those collectors were, perhaps caused the bottleneck themselves. Regardless, we used targeted capture to pull out these rad loci. And we also developed probes for the full mitochondrial genome. And we use this on the pool of DNA extracted from each bone sample. And then we also did capture of contemporary samples to get at the full mitochondrial genome from those as well. In the end, our data set from all of this was about 2,000 SNPs genotyped in 25 of the historical individuals. and then full mitochondrial genomes from 77 of the 78 historical samples. So we felt pretty good about this. In hindsight, if we had done a bit more sequencing, we would have been able to increase that sample size from the 25 individuals quite a lot. It was really just a matter of needing more depth, but so is hindsight. So we can compare directly now the levels of variation in the historical and contemporary populations. We can also subdivide the contemporary population to look at the different sort of eras of the Head Start program. It had been going on for more than 50 years, and record keeping is not great for this. We know roughly how many individuals were Head Started in each year, but we don't know how many nests they were sampled from, where on the island the nests were taken, that kind of thing. So it's possible that over time, with different sort of people leading the Head Starting program, there could have been a shift in behavior that would change how genetic diversity was being captured in those Head Start cohorts. So we divided up our contemporary population to adults that have a curved carapace length over 65 centimeters, and young individuals, so those smaller than 35 centimeters. So we're leaving out that intermediary group just to sort of get at earlier and more recent periods of the Head Start program. So we're looking here at heterozygosity and GIS, the inbreeding coefficient. So looking at our 25 historical samples and our 149 contemporary, we're not seeing any difference in these two measures. We're also not seeing any difference between the adult and young groups of our contemporary population. Another thing we looked at is whether patterns of relatedness have become skewed. And this is trying to get at this because all recruitment has been mediated through the Head Start program. Has this caused a shift in sort of the genetic structure at a finer scale? So the plot here is a distribution of pairwise relatedness values among individuals in our contemporary and among individuals in the historical data set. And this sort of distribution is centered right around zero relatedness. And these distributions are really largely overlapping, showing that there doesn't seem to be a difference in the overall structure of relatedness in the population. We can also look at the same thing between these early and later periods of the Head Start program. So in red there, it's the among adult relatedness distribution, and in blue is among young. So just looking at young individuals related to other young individuals. And you can see that there's sort of this bump towards mid-levels of relatedness. And it's our sort of explanation for this is that the cohorts in more recent years have been getting much larger. And so the cohort in 2009, which had been repatriated, that was the most recent cohort to be repatriated back to the island that we could sample there. It was that 2009 cohort was the largest ever at over 100 individuals. And so it's thought in that sampling, we managed to pick up more related individuals. And that's why there's sort of that bump in relatedness. But overall, we're not seeing a lot of differences there. We can then turn to our mitochondrial data to try and assess the genetic consequences of this bottleneck, because we know the population was reduced to about 150 individuals, which would have been the parents of these Head Start generations. We can also took this opportunity to assess the differences in inferences we gained from single mitochondrial genes versus the full mitochondrial genome. So this idea that with museum specimens, we're really only starting to get into genomics, and particularly, this was a number of years ago we published this. And so there was this sort of question in my mind anyway of, what are we getting from doing this full mitochondrial genome, this capture, this whole rigmarole, versus sequencing these mitochondrial genes, which would be more straightforward to do. And what you'll see here is so I'm showing, this is the haplotypic diversity of the historical population and the contemporary. So the historical is in blue. And keep in mind, our sample size for the historical here is 77 individuals. So this really is a high level of sampling. And then the contemporary, it's 50 individuals, so somewhat lower, but haplotypic diversity is corrected for sample size. So you can directly compare these values. So if you just focus here on these bars, this is the mitochondrial control region, so very commonly sequenced. And if you look at this, there is higher haplotypic diversity in the historical versus the contemporary. That trend is even stronger if you look at the CO1 locus, somewhat less strong if you look at cytochrome B, where we've got overlapping standard deviations there. So this is really showing that, if we had just sequenced the control region or the CO1, what our conclusions would be. But we do also have the full sequence, so we can look at that. And lo and behold, if you look at that full mitochondrial genome, there is no difference in haplotypic diversity between these temporal samples. And so our inferences really are influenced by our specific choice of gene in this situation. So to sum up the impacts of the Head Start program, since we found that the diversity statistics are roughly equal for the adult and young subsets, as well as they're similar overall to the historical population, it seems that Pinzon tortoises have maintained as much genetic diversity as was possible, given the bottleneck. And this is likely thanks to the quick rebound in population size enabled by the Head Start program. And so there's some lessons here, I think, that we can take away when we're talking about temporal studies in general. And the first is that looking at single or even a few mitochondrial genes could lead us to be either over or underestimating the differences in diversity compared to a larger sample of the genome. So this we found, temporally. So we might have concluded that, oh yeah, there was lots of diversity lost between the historical and contemporary population, when there hadn't really once you sequence the full mitochondrial genome. But this may also have implications for spatial patterns. So if we had sampled, spatially separated populations instead of temporally separated. And I think this gets into part of what Debbie was talking about a few weeks ago, that we really have to be careful about our markers and, pulling inferences from our systems, because it may not be telling the full story. Carrying on though, the other really important lesson here is generation time. So although our samples were separated by more than 100 years, in reality, this probably represented maybe one tortoise generation. So because there had been this pause in recruitment up until about the 1960s, where it resumes through the Head Start program, it means that there hadn't actually been time for genetic patterns to develop, nor had this bottleneck really had genetic consequences at that point. So it's really important, and again, highlighting some things from Debbie's talk, that we need to be talking about generation times, not years apart of sampling, especially in long-lived critters. But then also the impact of overlapping generations means that our expectations for how genetic variation will be impacted, we have to develop different expectations for that. So I'm going to move on to some other stories and some other things that we're learning from museum samples of Galapagos tortoise. So I'm going to switch now to talking about the tortoises on Volcano Wolf. So Volcano Wolf is that northernmost island, or northernmost volcano on Isabella Island. And the tortoises there are typically dome-shaped. But it was found that there are actually some individuals that have a saddleback shape. And they're so radically different looking than the dome tortoises that they're actually referred to in earlier papers as aliens, that they just so clearly did not belong in this particular ecosystem. But it turns out, thanks to some genetic analysis, that these are not aliens from another planet, but they're tortoises from a different island. And so it's through genetic analysis that it was found that these aliens belong to some of the extinct species of Galapagos tortoise. And we could only learn this taking advantage of museum samples of these extinct species as a reference that could be used to assign these aliens back to. So before, in the earlier years, before they had sequenced the museum samples, all they knew is we've got these weird mitochondrial haplotypes, and they don't really fit in with the genetic clusters that we see from the living populations. So it really was this bingo once they had the microsatellite data from these museum samples, that all of a sudden you could start assigning ancestry to these populations. And so up on Volcano Wolf, it turns out that there are hybrids of not one, but two extinct species. So the species from Pinzon Island, which in the bar plot is green, and Floriana Island, which is purple. And so in 2008, there was a huge sampling effort. They took blood from over 1,600 tortoises on Volcano Wolf, amplified them all at microsatellites, and did structure to reconstruct the ancestry. And so, sure, many of you are familiar, but the way to read this is that each bar is an individual, each color is a genetic cluster, and so the portion of each bar that's each color is the portion of ancestry. So you'll notice that a lot of purple in this plot. So although they didn't find any pure individuals of either Floriana or Pinta, the extinct species, there's a whole lot of hybrids. And so this sort of presented a tantalizing opportunity for the Galapagos. Floriana and Pinta Islands have no more tortoises left, and the tortoises are really important ecosystem engineers. They're the dominant herbivore. And so having tortoises back on these islands is really an important part of their ecological restoration. So a plan was developed to take these hybrids from Volcano Wolf and breed them in captivity to produce individuals that could be used to repopulate those islands that have at least some ancestry from the original species. So they're not trying to like rebreed them back, the extinct species, because that's just not possible. It also would be like a 500-year project with tortoises. We're talking about, a single generation in captivity to breed individuals for release. And then having all of this very closely monitored using genetic markers. And so that's what's happened in 2015, and then again in 2020. There were massive expeditions to Volcano Island to find these individuals that had been previously sampled and identified as hybrids, and bringing them into captivity to found a captive breeding program. And so this is a massive undertaking. So there were more than a dozen teams of rangers out combing this very rough terrain of basically an active volcano with all the loose rubble and thorny bushes and ticks like you wouldn't believe. I didn't get to participate, unfortunately. But finding these tortoises, calling the helicopter, having them roped up and then taken back to the boat where they sat for a few days before they were brought into captivity. Again, showing the remarkable dedication to restoring these tortoises. So there are now more than 40 of these Floriana hybrids in captivity, and the breeding program has begun. And so this program is currently informed using microsatellites. So both for identifying the breeders, so identifying their hybrid ancestry, as well as figuring out who should be breeding with who, assessing paternity of clutches. All these things are being done with microsatellites, being very closely monitored. But we'd really like to change this program to be using SNPs for many reasons, one of which the facilities for genotyping microsatellites are diminishing. And we know, right, that it's best conducted in the same lab over time. And that's becoming challenging with microsatellites. So changing to SNPs makes it less or more agnostic to, which lab is doing the genotyping. Of course, these markers provide more resolution, so we can minimize inbreeding while maximizing the amount of Floriana ancestry. And there's another important part of this that it actually took me a long time to really realize, like what is really a good reason for switching to SNPs. And it's really to reassess ancestry in higher resolution. It was a panel of 12 microsatellites that were used to identify, genotype first in the museum specimens to come up with the reference populations, and then identify hybrid individuals. But tortoises have something like 50 chromosomes. And so we're nowhere near even one microsatellite marker per chromosome. And so we could be completely misassessing the proportion of ancestry just based on, the random sampling of where our microsatellites are. So lots of good reasons to switch from using microsatellites to SNPs. The first step in this, of course, though, is to develop markers for the Floriana species. And unfortunately, this has now become the long struggle for a Floriana genome. We've been facing a lot of difficulties with degraded DNA. The specimens that were used as references for the microsatellite work are hugely degraded samples. So these are bones that were found from naturally deceased tortoises that were actually in a cave on the island. So these tortoises deceased at some point due to natural causes, probably falling into the cave and then starving. And so they are really tricky samples to work with. And so we've gone on this long hunt to find a Floriana specimen that was collected live. And this leads to the problem in finding specimens that are actually from Floriana, because the island actually had a processing facility that slaughtered tortoises to get their oil. Tortoise oil was used, it was exported and used for like fueling street lamps. So you can imagine street lamps being fueled by Galapagos tortoise oil. So even if a museum record says a tortoise was collected from Floriana, in many cases, it was an individual brought from another island to this processing plant that then, sailors or collectors picked up. So this has been a very difficult problem to deal with, is finding a high enough quality specimen that is actually from Floriana. We have looked at many tortoises that ended up not being from Floriana. I have one more story I'm going to tell before I wrap up here. And this is the story of the tortoises on San Cristobal Island. So San Cristobal is over in the easternmost part. It was one of the first islands colonized by the tortoises. And it was, heavily exploited like elsewhere. And so by the 1930s, tortoises were extirpated from all over the island, except for the very northernmost part, where about 700 individuals survived. And we know that they've experienced a relatively severe bottleneck because they are actually fixed for a single mitochondrial haplotype. Our friends, the California Academy, back in 1906, they surveyed the island and they actually only found one living tortoise. And this is because they were looking in the southern part of the island and they were largely extirpated to the north at this point, up to Punta Pit. So they found this one live specimen about in the middle of the island there. And they also found this cave that had the bones of tortoises that had fallen in and naturally deceased. And they found no other tortoises. And we got access to these specimens with the goal of being able to do a temporal study to look at what has happened in the San Cristobal population, how it ended up, being fixed for a single haplotype, and just sort of look at the patterns there. So we have bones from 5 of the cave specimens, as well as the individual that was collected in 1906. And we were going to look at temporal patterns of diversity. So for this, we sequenced the mitochondrial control region, and then we tried really hard to get microsatellites from the museum specimens. And this all failed. Those bones are in really rough shape. but at least we were successful getting the control region. So I'm showing a phylogeny here of all of the living lineages and some of the extinct ones, but we're just going to zoom in down over here. And so in purple, I've highlighted this is the haplotype that's found in the contemporary population. And the individual that was sampled live in 1906 has a haplotype that's very closely related. But to our great surprise, those specimens from the cave form a clade that is sister to the tortoises on Hispaniola, so a completely different island, and are not at all closely related to the other San Cristobal haplotype. I mean, this is pretty exciting. It looks like we have found a new lineage of Galapagos tortoise just based on these museum specimens, sort of in a place we weren't really looking to find another lineage. So we went ahead and carbon dated one of the cave specimens, and it came back between 1450 and 1630. And so this suggests that the lineages were actually likely sympatric on the island, given the temporal proximity here, where if this tortoise deceased, sometime around 1600, and then 300 years later, they're sampling a live individual very close by. But not having any nuclear data really does make it difficult to make statements about what are these specimens really. We've just got a fragment of the mitochondrial control region, and it seems to be a completely separate lineage. And so we did what we could, which is to closely examine the contemporary population for any trace of this lineage. And so we dug in and we sampled more than 240 of these living individuals and sequenced their control region. And yet we still find that there is only this one mitochondrial haplotype. So they really do seem to be fixed at the control region. We also looked at 64 individuals at 21 microsatellites, as well as more than 13,000 rad loci. And we find no evidence that there's any sort of substructure or gene flow with any lineages. There's no patterns here that would link up to these cave specimens. So our next step on this is working hard to get whole genome sequences from these cave specimens. And so learning from our experience with the Floriana, shotgun sequencing has really been unsuccessful due to the low endogenous content of these old degraded bones. And so what we've turned to is using whole genome enrichment to try and increase the proportion of tortoise DNA in what we're sequencing. So meanwhile, the search continues for representatives from Floriana. And to our surprise, when we're screening all these potentially Floriana specimens, we find two that are of unknown provenance. We don't know where they came from, but they have mitochondrial DNA that's similar to these cave specimens. And one of these we were able to obtain a whole genome sequence from at sort of relatively good coverage. And it comes up as being sister to the clade of the contemporary San Cristobal tortoises. So this is sort of a tantalizing teaser that, even though mitochondrially, they're completely different parts of the tree, based on our whole genome sort of nuclear SNPs, they are nesting right in there with the rest of the San Cristobal tortoises. So we're now working with anthropologists to try and trace the provenance of these specimens. So that's looking back through ships' logbooks, donation records for natural history museums, all these things. And we are setting up a new ancient DNA lab at Newcastle, which is making it easier for us to analyze specimens from museums in Europe that, due to difficulties with, CITES and permits and shipping things internationally, have largely been unexplored, the Galapagos tortoises. So continuing on for the lessons for temporal studies, I just want to highlight that there are some really exciting surprises to be found in old specimens, even where you're not necessarily looking for them. And Galapagos tortoises being, very much charismatic megafauna, they have been relatively well studied. And yet we're finding these unexpected new lineages. So what other surprises are just sitting on the shelf there for other taxa? I also will emphasize it's very important to not believe what's written on the tag at the museum. It could be anything, but that's part of the excitement, is to try and trace down the history and identify those specimens. So I'll just wrap up by thanking the California Academy of Sciences and the Museum of Comparative Zoology at Harvard, where we sampled the specimens that I've talked about today. And of course, the Galapagos National Park, who are partners and collaborators in the work on the tortoises. Also have a number of funding sources, and I just want to quickly highlight the American Genetic Association. So there's a grant that they give out each year that can be used for genomic studies. And it's available for either PhD students or postdocs, and it's up to something like $10,000. So if you're a PhD student or a postdoc who would like some money to do some fun genomics work, I highly suggest that you look into that. So I'll just finally thank the Temporal Genomics Working Group for organizing this seminar series. I've really been enjoying it, and I am very happy to have had a reason to talk about tortoises this afternoon. So thank you.
Great, thank you, Evelyn. Cool. Yeah, I'm wondering, I haven't seen any questions in the chat yet, but, and I haven't seen any hands raised yet. I can just ask a quick question. I thought those cave specimens are really interesting. Can you tell at all from the bones if the morphology was similar to the contemporary surviving turtles, or was it, is it possible that these were, morphologically similar in some way, that they would be competing, or did they have that kind of difference in, like, some saddleback, some domed?
It's really hard to tell. So the bone fragments are in bad shape. There are some carapace, and they seem to be sort of saddleback shaped, whereas the specimen that was collected alive in 1906 is very much domed. But that individual is a sort of a small female. She's only got 3 legs. She lost one due to an accident. So it's possible that she's not necessarily a morphologically typical individual. And then the contemporary population on San Cristobal actually has an intermediate shell shape between domed and saddleback. So it seems like there could be some sort of interesting complex story there. And yet somehow they have ended up completely fixed at their mitochondrial genome. So, yeah.
Cool. Awesome. Alexa has a question. Alexa, do you want to ask?
Sure. This was kind of long to type in the chat. Thank you so much for the amazing talk. This isn't a genomics question. It's just about the tortoises. I was so curious when you were talking about this captive breeding program, like, where is it? Is there just some giant park somewhere where they're rearing tortoises? Like, how durable do you think that program is? Where is its funding from? How do you see, like, the conservation outlook for these species in the context of, like you said, the fact that most of the adults are raised in captivity by humans?
Yeah, so great question. The captive breeding program is in situ. It's in the Galapagos. There are the largest one is on San Cristobal Island. So this is the island that has the most human development. That's where the Charles Darwin Research Station is. And so that's where the captive breeding and head starting has been based out of, over all these decades. They've now set up a separate facility on Isabella Island, just sort of redundancy and extra capacity. So these captive breeding programs have been hugely successful. They've been operating since the 70s. They have saved the Espanola tortoise, which was down to 15 individuals. And they have captive bred these individuals, genetically monitoring, like doing it right. And that species now numbers over 2,000 in the wild and is reproducing in the wild. So these programs are really remarkable, really successful. They are also run on a shoestring. these, this is in the Galapagos. And, these are rock walls, enclosures. So they're really the best case scenario captive breeding program in that it's in the environment, it's being genetically monitored. They're repatriating tortoises. These are single generation captive breeding programs where the offspring, as soon as they're big enough to survive out there, they're being repatriated. They really are remarkable programs. And yeah, just the dedication of the rangers and the national park and keeping those running. But with COVID and the huge, a lot of the money in the Galapagos comes from tourists, all of the money basically. And so the impact of basically not having tourists for a year is really uncertain. So if anybody knows somebody with a huge pot of money that wants to put it towards something really good, finding these captive breeding programs this is just the tortoises I'm talking about. There's other iguanas and things in the Galapagos they're doing similarly good work with. But yeah, they're running on nothing, especially right now. And yet they do such good work.
I think Leonardo DiCaprio is a big turtle conservation donor, so you might.
Oh, well, if anybody's got his number.
Yeah, I don't, but I hear things. Okay, let's do, so we have a couple hands raised. Let's do John, then Malin, then Mike Hickox. So John, you can go first.
Hey, Evelyn, great talk. So a lot of harvested populations have reduced sizes over time. But since the smaller individuals from Pinzon were preferentially harvested, was the contemporary population larger on average than the historical population that you compared it to? And did you identify any candidate loci of harvesting that might be associated with morphology or size?
Yeah. So the tortoises on Pinzon, there's not so much that they were size selected. They were selected as a species. This was an easy island to go to get tortoises compared to the other ones where, being small and arid, it's relatively easy to traverse. And then you can just pick up one tortoise. So that's where the sort of size selection comes in, is that they were just heavily harvested compared to the larger species. And then, yeah, we didn't find any shifts in sort of total genetic variation between the historical and contemporary. And so, yeah, whether there's been any sort of meaningful evolutionary shifts, we don't have any data to support that. But also, this is really just one generation.
Right.
So.
All right, Malin, you're up next.
Yeah. Hi, Evelyn. Thanks so much. That was it's quite a system and a really cool talk you gave. I was curious, it seems like you're really pushing for SNP data, whole genome data, which makes a lot of sense. It seems like that would open up, if you were able to do whole genome or at least subgenome sequencing with these historical samples, you'd open up a whole nother range of really cool research questions. So I was wondering if you could just talk a little bit about what seems to be working maybe, or what you think might work in terms of making progress on that. It sounds like you're sort of at the technical lab side, which makes a lot of sense, right? It sounds like maybe DNA preservation has not been very good. Makes sense, it's near the tropics, but.
Yeah, so we've taken sort of a number of ways. So, one way to improve your data is to get better DNA out of your bones. And so to tackle that, we've really optimized our extraction protocol from bones, but also I started CT scanning all of the bones and using that to select the densest region of bone and very specifically targeting that for DNA extraction. And that's made a big difference. So our intuition, sort of as a research group of, you looking at this bone, where should you, take a piece from, was not, our intuition was not targeting the most dense part. And so having now looked at those CT scans and learning about that has improved things a lot. The other side is that, you can, preparing libraries like DNA sequencing libraries, our kits are so good right now. That's not a problem. You can do a single stranded library prep from these ancient samples and make sequencing libraries. But when you have less than 1% of your target species DNA, you can sequence forever and it will never add up to much. So one specimen I have more than 800 million reads for, and I don't even have a 5x mitochondrial genome. So like my entire PhD was like on a fraction that number of reads. So this isn't a problem you can just like sequence your way out of. So that's where this enrichment, I think, really comes into play. So we had done it with the Pinzon tortoises using this capture just to get at the rad loci. And that was successful. And if we had done more sequencing, I think it would have been even more successful, right? If we had looked at the data and known and then gone and done more sequencing. So that's where we are right now with the whole genome, is doing whole genome enrichment, which is where you take DNA from a modern individual and it, through magic, gets turned into RNA baits. So this isn't a synthesis. This is like things happen in a tube. It's not synthesized by a machine, which other capture approaches are. So like our targeted capture with the rad tags, that was synthesized. This is contemporary DNA being turned into capture baits. And so it can be done for any species, right? You don't need to have existing genome resources to do it. And then using those baits to enrich your prepared library for your target species. And honestly, I was pestering the sequencing center today, being like, where are my sequences? Because they have them from this first test of the whole genome capture. So I'm really hopeful, and that's got to be the only way to do it. And it's not as expensive as we thought. So I could have saved money by going to this capture approach in the first place instead of sequencing to 800 million reads one sample, and still never going to get anything from it. So anyway, I'm happy to talk technical stuff to anybody that wants to hear about it.
It's super helpful to hear your experience. It's a common challenge with historical DNA.
Okay, MK, I'm very sorry. I think I misnamed you. I think I called you Mike before, but MK, and then Debbie will be next.
No worries. Evelyn, thanks so much for the talk. It was really cool to like see everything you're up to now. So I have two questions. The first one, I was wondering, how have you been identifying the potential Floriana samples? Like, are you able to do nuclear, or is that through mitochondrial DNA? And then I was also wondering if it's possible at all to get any samples from private collectors, because obviously, like, the tortoises were spread out a lot and like exported, but then also I know it's kind of like a sketchy business and people might not want to like actually admit that they have tortoise shells.
Yeah, so I was listening. What was the first part of your question again? I'm sorry. Oh, how we're identifying them. Yeah, museum records. So you do have to start somewhere. And so it starts with what's the label on the sample. And then we're extracting DNA. We're doing Sanger sequencing of the mitochondrial control region just to see, what lineage does it fall into. And the good thing with the Galapagos tortoises is they are quite differentiated. And so based on the mitochondrial control region, you can assign it roughly to which species and which island. Where we run into trouble is the ones that the Sanger sequencing doesn't work very well, but maybe that's because they're really degraded and the provenance seems good. And so we sequence and never get anything, or they do end up being the wrong species. So we're now, teamed up with an archaeologist whose passion really is reading old logbooks of expeditions to the Galapagos. And he's been so helppful in identifying specimens in museums around the world. So we've got all sorts of requests out for, the critters I've talked about today, but also there's every island in the Galapagos has a fascinating story that I could have talked about. So that's been really helpful. And then, private collections, it's hard to know if anyone's going to admit to having a tortoise. And then if they do, how good is their provenance? But thanks to this new collaboration with an archaeologist, anthropologist, I've got leads that I'm very excited about.
Cool. Okay, Debbie, you're up next.
Thanks, Ev, for such a great talk. I had a question that was kind of inspired by some of the post-hoc discussions that we just had. I think it was John's comment about size selection in harvesting. In any of the bottleneck species, especially this one where you only see one haplotype, are you also looking to see if the Y chromosome is similarly homogeneous? Or
What
is it,
Y
or what, how
chromosome?
is it, what is it in tortoises? How are they sex
They are
determined?
temperature dependent sex
Oh, yeah,
determination.
okay, so never mind then, that was.
So there are no genetic markers for
Mhm.
sex. Yeah,
Okay, so then you can't do that. And that okay.
yeah. Well, which is its own interesting problem, you know, from
Yeah.
a conservation perspective, is that one, well, so if you're managing these captive breeding programs, you actually have control over sex ratios. And so that goes into all these simulations we're running for what is the best way to manage a captive breeding program.
Mhm. Well, thanks. I forgot that they were temperature. I feel like such a geneticist now, like forgetting the life history of a species.
No, it's just one of those that because they're tortoises. Yeah.
Yeah.
Turtles are cool and weird. That's my final thought, unless anyone else has any questions for Evie.
Yeah, anyway, if any of you send me an email or on Twitter or anything, I'm happy to talk technical details. I love talking about other people's projects. So yeah.
Great. Thanks, Evie, and thanks everyone for coming. And remember, next week we have a quadruple header. We'll have 4 different early career researchers giving summaries of their work. So that should be really neat. Okay, great. So hope everyone, yeah, that's 2 weeks from now, 2 weeks from now. So yeah, see everyone hopefully then.